So we're going to wait just a little while longer while people get settled, but we'll be starting very soon. I think we're there, so let's begin. I'm Ward Holder. I'm the director of the honors program at St. Anselm College, and it's my distinct pleasure to introduce our speaker tonight. In fact, for me, this is a singular pleasure because I've never had the opportunity to say this before. Professor Jennifer Pace is an assistant professor of chemistry at St. Anselm College, but this is the first time we've had a speaker in our lecture series. The first time we've had a speaker in our lecture series that I had as a student in class. So it's special for me. Prior to coming to St. Anselm to teach, Professor Pace was a postdoctoral scholar working with Joshua Kritzer at Tufts University. Professor Pace not only teaches in the chemistry department of St. Anselm College, but is also teaching a course in the biology department this semester she took while she was a student at the college. After her Bachelor of Arts in Biochemistry from St. Anselm, she did her doctoral work in chemistry at the University of Connecticut, working under the direction of Professor Kyle Haddon. Professor Pace is widely published in the broad field of anti-cancer chemotherapeutics. She has published articles in an impressive array of journals that represent the cutting edge of biochemical research, such as Chem Biochem, the Journal of Liposome Research, the Journal of Medical Chemistry, Tetrahedron Letters, and the Current Medicinal Chemistry. She has been funded by New Hampshire INBRI to continue this work at St. Anselm College with her students. Professor Pace has agreed to take questions at the end of her lecture, so we encourage you to use the Q&A function and write down any questions that you might have. Tonight, Professor Pace's lecture is entitled, "The Race Towards Finding a Treatment for COVID-19." Welcome, Professor Pace.
All right, I'm going to share my screen. All right. Hi, everyone. Thank you, Professor Holder, for that kind introduction. And thank you to the rest of the Honors Council for inviting me to speak during this unique running of the Honors Lecture Series. I'm very excited to be here today on Zoom, and I hope to provide an informative lecture regarding the science behind COVID-19, as well as the scientific research associated with discovering a treatment for this debilitating disease. So far, Professor McLeod gave us an insight into the effects that COVID-19 is having on our frontline workers, especially nurses. Personally, hearing these stories from our own St. Anselm alumni who are caring for COVID patients and essentially dedicating their lives to the pandemic was really truly eye-opening. Through this week's lecture, I will provide a scientific overview of COVID-19 that will serve you through the continuation of this honors lecture series. You have already heard how COVID-19 has affected nursing. And next, you will hear about how it is affecting politics from Professor Lucas. COVID-19 has clearly affected all aspects of our world, but why? What makes this virus so different than others? Before I jump into the science, I really wanted to reiterate that we knew next to nothing about this virus at the beginning of the year. From determining where COVID-19 binds in our bodies to how the virus is transmitted between humans, everything we now know about the virus can be attributed to ongoing rapid scientific research. There are many scientists in research laboratories that do not normally study viruses that ultimately switched gears to help us all learn more about COVID-19. With that, scientific research articles surrounding COVID-19 are being published at an increased rate. Scientific journals are struggling to balance their traditionally slow and careful approach with the desperate demands of society seeking to save lives. With this, scientific research is becoming more open, more efficient, and more collaborative. However, the downside to this accelerated release of information is that sometimes it proves to be wrong. Some high-profile COVID-19 papers have been released, promoted by the media, and ultimately retracted. Additionally, COVID-19 pandemic has caused a decline of research in other disease areas. And this is due to just the generalized focus on COVID-19, but it is also due to our lockdowns and the inability for some of these researchers to get into the lab. So with that, I'm going to jump into the science. We're going to first start by talking about what a virus actually is. A virus is a small infectious agent. It's ultimately smaller than both fungi and bacteria. It can only live within a host and therefore must invade a living cell to reproduce. In order to reproduce, viruses release their genetic information. Ultimately, the virus takes control over the host cell, and that host cell is able to produce more viruses. As a result, the host experiences symptoms of disease. And some well-known viruses include rhinovirus, or the common cold, influenza, which is the flu, and neurovirus, which is an awful stomach bug. I think one thing that gets misinterpreted within the media is the back and forth usage of coronavirus and COVID-19. And it's something that I even have to stop and think about myself. Coronavirus is a family of viruses that typically cause cold-like and respiratory symptoms. Some other examples of coronavirus include severe acute respiratory syndrome coronavirus, or SARS-CoV. There was an outbreak of SARS-CoV back in 2003. And Middle East respiratory syndrome coronavirus, or MERS-CoV, which caused an outbreak in 2012. Ultimately, coronaviruses originate in animals and sometimes leap to humans. In most cases, they start in a bat, transfer to another animal, and then ultimately to humans. In the case of COVID-19, there are mixed reports on this transmission, either being directly from a bat or through this intermediate host known as the pangolin, which is an anteater-like animal that lives in China. So how does COVID-19 work? COVID-19 has spike proteins that protrude from its surface. The virus is also enveloped in a bubble of oily lipid molecules. This bubble ultimately falls apart with soap, which is why everyone is telling you to wash your hands. The virus enters the body through the nose, mouth, or eyes, and then attaches to cells in the airway that produce the protein ACE2. Once bound to ACE2 and into the cell begins to form more copies of COVID-19, which are then brought to the outer edge of the cell. Each infected cell can release millions of copies of virus before that cell dies. Virus that is released can either infect a new cell or it can be released as droplets from the lungs. As of now, COVID-19 appears to be so contagious because we have never been exposed to the virus before. The R0 scale represents the average number of people that one person with the virus infects. So in terms of COVID-19, it has an R0 value of 2 to 2.5, meaning that one person infects 2 to 2.5 other people. Controlling this transmission is necessary, and this can be accomplished through social distancing and mask wearing. However, as of date, COVID-19 has claimed the lives of 1 million people, and we need a treatment, and we need it fast. So what kind of treatments can we utilize to stop the symptoms of COVID-19 or ultimately prevent the symptoms from getting as bad as they are? I'm going to switch gears a little bit and start by talking about what drugs actually are. When we think of a drug, most of us think of medication that we either pick up at Target or at the pharmacy. But in reality, drugs are considered to be any compounds which interact with a biological system and produce a biological response. For instance, morphine interacts with the body to bring pain relief. Snake venom interacts with the body to cause paralysis or death. LSD interacts with the body to produce hallucinations. Penicillin interacts with bacteria to kill them. And one of our favorites, caffeine, interacts with the body to wake you up. Now, drugs come in a variety of shapes and sizes, from small molecules like aspirin to larger macromolecules like antibodies or genetic components of vaccines. The size of a drug has pros and cons. For instance, smaller drugs are more easily made in the lab, while larger molecules are more difficult to synthesize. It is important to explore this broad array of drugs for a particular disease or ailment because it's hard to predict what type of drug might be more effective. So when we think of a treatment for a virus, most of us have probably had the experience where we go to the doctor or to urgent care and we might be told, go home, rest, maybe take some Dayquil. There's not really a ton going on for viruses that we would typically use in terms of treating a cold. However, there are two main pharmacological ways that we can deal with viruses. The first is preventative. Vaccines are able to train your immune system to create antibodies to fight off a virus. They are important towards preventing and controlling disease outbreaks, and they are actually available for over 20 life-threatening diseases. The second treatment option are antivirals. Antivirals can be either specific to one virus or they can be focused on a broad spectrum of viruses. Antiviral drugs do not destroy the target pathogen, but instead prevent the virus from reproducing. The only issue is that viruses can eventually build up resistance towards these compounds. So that means that if you were taking an antiviral on kind of a regular schedule, it may not work as effectively. So this is an infographic that highlights just some of the research regarding COVID-19 treatment. So the Milken Institute has collected information in terms of this COVID-19 treatment research pipeline. And the awesome thing about this website is that you can actually go in and look at all the data and make your own graphs, which I think is pretty cool. So as you can see, there's a lot of research going on, ranging from vaccines to antivirals and a few other types of therapeutics that we could utilize. Now, I am aware that this infographic is for March. I did try to go on and play with the data. And now, since it's October, there's a lot more going on in this graph, and it was very overwhelming. So I just stuck with the one for March. Ultimately, we are dealing with a virus that we literally knew nothing about a year ago. How feasible is it for us to have a treatment or preventative within the next few months? So this is the drug development pipeline, and it typically takes about 12 to 15 years and costs more than $15 million to get a drug FDA approved. Clearly, in this situation, we don't really have that amount of time to work with. People are dying in mass amounts, and we need to find a treatment to combat this disease. As you can see in the pipeline, the initial beginning stages of the drug development process consist of early stage research and development. So we're finding our chemical structures, we're testing them in cells, and the second half is about clinical trials. And this is when we're checking the drug safety, its dosage, and its efficacy in humans. What are some ways that we can bypass this timely and expensive process and arrive at some sort of treatment for the time being? One way we can expedite the drug discovery process is by making use of pharmaceuticals that are already FDA approved. This is known as drug repurposing or drug repositioning. Drugs that are FDA approved already have been deemed safe in clinical trials, and we already know about their potential side effects and potential drug interactions. In drug repurposing, it is of particular interest when we are dealing with some sort of disease outbreak, so COVID-19 pandemic. We also saw this in terms of the Ebola and the Zika outbreaks a few years back. This is also of particular interest when we're looking for a treatment for an orphan disease, and orphan diseases are considered to be diseases that really only affect a small subset of people. Most drug repurposing actually happens serendipitously. So the oldest example of drug repurposing is actually aspirin. Aspirin was originally marketed in 1899 to treat pain. However, it was repurposed at low doses as an antiplatelet aggregation drug, which ultimately prevents blood clots and helps prevent cardiac events. Thalidomide was originally marketed as an anti-nausea medication for morning sickness. It was banned by the World Health Organization in 1962 due to birth defects. However, it was reapproved in 1998 for the treatment of leprosy and skin ailments. So with this in mind, a study was performed in which 12,000 FDA-approved compounds or compounds that are currently in clinical trials were explored for their ability to inhibit the reproduction of COVID-19. From this initial study, 21 of these drugs proved promising and have been further studied for their novel biological activities. Since this initial study, we have learned a lot more about these 21 drugs in terms of their effects on COVID-19. In the interest of time, I have decided to highlight the 4 main drugs that most of us have probably heard about on some level, on either social media or through the news. So the first that I'm going to talk about is hydroxychloroquine and chloroquine. Both of these compounds are FDA approved for the treatment of malaria. At the beginning of the pandemic, both of these compounds proved promising as they inhibited COVID-19 in cells. However, upon exploration in both animal models and randomized clinical trials, it was determined that both of these compounds actually weren't beneficial to COVID-19 patients. In fact, they were actually pretty harmful to COVID-19 patients. Both of these compounds were also found to inhibit Ebola during the Ebola outbreak. However, they also didn't perform well in the animal models. So it's interesting in that we're seeing viral activity in cells, but not when we're putting them into an organism like an animal or a human. The next drugs that I'm going to talk about are lopinavir and ritonavir. This combination of drugs was approved to treat HIV. Initially, these drugs were found to stop the virus from replicating. However, clinical trials thus far have proved disappointing. Remdesivir exhibits its antiviral activity by inhibiting the creation of new viruses. A clinical trial proved promising results in that the drug reduced the recovery time of patients from 19 days in the hospital to only 11 to 15. This was the first drug to get emergency authorization from the FDA for use on COVID-19 patients. It was also originally explored for the activity against both Ebola and hepatitis C. And finally, dexamethasone is a type of steroid that is used to reduce inflammation seen in both allergies and asthma. In the 1960s, it was actually used to treat pneumonia and other respiratory conditions. A study concluded that dexamethasone and other steroids linked to 1/3 reduction in deaths in COVID-19 patients. So, now that I've kind of introduced where we're at in terms of small molecule drug development and antivirals, we're going to move on and talk a little bit more about vaccines. So I'm going to talk a little bit about how vaccines work, the different types of vaccines, and also the current vaccine development landscape for COVID-19. So we're going to start off by talking about how vaccines actually work. Typically, vaccines consist of either weakened or dead material that is introduced to a patient. The immune system triggers a response to fight the disease. You will probably hear people say something like, we're producing antibodies. That means that our body's fighting the disease. If the patient encounters the disease later in life, it is easier for them to develop antibodies and for their body to ultimately fight the disease. So vaccine development typically requires 10 years of research and testing before reaching the clinic. But scientists are racing to produce some sort of safe and effective vaccine by 2021. Every disease has different characteristics, so each vaccine will have an individual development path. Depending on the disease, pathogens are usually either reduced in potency or inactivated. Before any testing in humans, a new vaccine must undergo careful investigation in the lab, followed by trials in animals. We must confirm that the vaccine is signaling the immune system appropriately. Testing in humans is conducted in 3 different phases. 1 to evaluate the safety, 1 to evaluate doses, and 1 to evaluate protection. Ultimately, clinical trials take many years to complete. So when scientists are developing a vaccine, they're thinking about a few things. 1, how our immune system responds to the virus, who needs to be vaccinated against the virus, and the best technology and approach to create the virus. To start, there are several different types of vaccines. Each type is designed to teach your immune system to fight off certain types of virus. The 5 types I have shown on this slide are really the 5 types of viruses or vaccines that are being explored for COVID-19. The first is genetic vaccines. These deliver one or more of the coronavirus's own genetic material into our cells to provoke an immune response. And an example of this is the vaccine developed by Moderna that has been in the news a lot. This is currently in phase 3 clinical trials. And this final trial actually involves enrolling 30,000 healthy individuals at about 89 different sites in the United States. We have viral vector vaccines, and these contain viruses that are engineered to have the coronavirus genes. Protein-based vaccines contain viruses that, or contain proteins, but not genetic material. The inactivated virus vaccines contain viruses that are weakened or that have been killed by chemicals. The last subset of vaccines are actually repurposed vaccines, so they are exploring a few vaccines that are utilized for other diseases for COVID-19. Ultimately, like molecules and macromolecules, different formulations of the virus have pros and cons regarding their safety and efficiency. So currently, there are actually 43 vaccines in clinical trials and at least 91 vaccines in preclinical trials, which means that they're under active investigation in animals. At the end of the day, there's going to need to be a strong cooperation between the vaccine developers, public health bodies, and government officials. These groups will all be needed to ensure promising late-stage vaccine candidates can be manufactured in sufficient quantities and also be supplied to all affected areas equitably. Taken together, the global research and development effort in response to the COVID-19 pandemic is unprecedented in terms of size and scale. Of all the drugs trialed so far, only steroids have been proven to save lives. However, dexamethasone does not work on people with mild symptoms. There are also new COVID-19 drugs that are in the pipeline that are currently being studied in lab. However, they are in the beginning stages of development and are not ready to be studied in humans yet. I want to reiterate that we may never have a cure for COVID-19. We don't have a cure for the flu, and we don't have a cure for the common cold. However, finding a treatment or preventative will ultimately save lives and allow for hopefully lifting some of the restrictions that we're seeing in terms of lockdown and social distancing. And with that, I'm happy to take questions, and I thank you all for coming here today over Zoom.
Jen, can you hear me?
Yeah,
So I have one for you.
okay.
Coronavirus.
Mhm.
I was fascinated that that these all start in. They tend to start in bats. Why?
That is a great question. I actually, I'm going to be honest, I don't 100% know. I'm going to assume it has something to do with the way that bats, like what they feed on and things, that they just carry different viruses that we typically wouldn't have. But Off the top of my head, I'm not 100% sure. That is a great question. Not seeing any questions. Oh, I have one. How much hope do you have that a vaccine will be ready by 2021? This is a tricky question. In my honest opinion, I think that there could be a vaccine, but I do think that I myself, and also including many other people, might be hesitant about that vaccine just because of how quickly it has been churned out. I think that's going to be, even if the vaccine is effective and seems safe, I think that's going to be the biggest hesitation amongst all populations of people. Here come the questions. Maybe there was a delay. If you had to guess, what kind of virus will be most effective against COVID-19? I'm guessing that that might mean vaccine or drug treatment. In terms of the vaccines, I think that's a toss-up. I think Moderna is a genetic vaccine, and that seems to be doing really well right now. Of the different types of vaccines, is one type preferred or considered safer? Yes. So all of those different vaccines, and I almost included it today, they range in terms of safety and efficacy. Do you think COVID-19 will impact policy changes towards research for antivirals and vaccines? Yes. I think that COVID-19 is going to put viruses and vaccines on a little bit of a pedestal in terms of other disease states. I also think that because the research has been proving to be going more rapidly, that might actually be something that scientists continue to stick with, and we keep moving a little bit faster than usual. How safe do you think a vaccine that comes out in the next year could possibly be? I think I already answered that a little bit earlier. I think there will be hesitation. I do trust my scientists and colleagues that hopefully it is safe. A lot of drugs in use are ones that are being repurposed from other treatments. Why were these particular ones chosen, and are there other newer ones in the pipeline? So these drugs were chosen. They were found to be basically hits in a previous study that tested a bunch of FDA-approved compounds. So I guess they weren't really chosen, but they kind of fell into the, I can't think of the word. They were productive in that assay. And are there newer ones in the pipeline? Yes, there are newer chemical entities that are being studied. So basically starting from scratch, not necessarily repurposed drugs. Is it important to get a flu shot this year because of COVID-19? Yes, please get the flu shot. When do you think we'll get back to normal when we're not having to social distance, or will this be the new normal since we don't have cures for cold or the flu? I think that's also kind of a loaded question. I think that we will be socially distancing for a little bit. I think that the more we are exposed to this virus, like the cold or the flu, we may see some changes. Again, this virus is so new, and our bodies aren't ready for it. So I think we might be doing this for a little bit. Do you think there will be multiple vaccines later, or will the first one be the main/only one used? I think there'll be more down the pipeline, absolutely. Do you think that scientists should consider not focusing so much on COVID-19 and continue their research on other diseases that are more likely to be cured? I think that at some point, scientists are probably going to fall back into where they originally studied. Right now, there's a lot of funding in terms of COVID-19, so I'm sure that is a big desire to some scientists and research labs. But all diseases are important, so I hope that we can also get back to looking at cancer and everything else. I think I got them all. Awesome. Well, thank you all for coming here tonight. It was a pleasure to be able to talk to you all. I hope to see you all at the next honors lecture seminar. I'll be there. You won't see me probably, but it will be on October 27th at 7 p.m. with Professor Lucas.